Hello, good morning everyone. Respected dignitaries on the dais, our esteemed chief guest of today's function, Professor Dr. Surendra Ghaskadbi, Vice Principal and Head of the Department, Dr. Dilip Kumar Balsingh sir, uh, Head of the Department of Microbiology, Dr. Avinash Tawde sir, Director of Biotechnology, Sunny sir, we, the teaching, non-teaching staff, and the students of Department of Zoology, Ahmednagar College, extend a warm and heartfelt welcome to all of you on this wonderful morning. Thank you for accepting our invitation to attend the function. To begin with the proceedings, I would now like to invite all the dignitaries to garland the portrait of our Reverend Founder, Dr. Bhaskar Pandurang Hiwale, as a mark of our respect and gratitude. Dr. Hiwale has always been our guiding force and inspiration.
Good morning, everyone. Respected dignitaries, Dr. Surendra Ghaskadbi, Head of the Department of Micro, Dr. Avinash Tawde sir, then Head of the Biotechnology Department, Dr. Sunny sir, my colleagues, Head of the departments, as well as my all dear friends. Today I am delighted to have a unique personality with us in our college campus, who have been the alumni of this particular college, as well as he was there in this college when the golden era of our institution was there as such. Now on this particular occasion, I want to say that he is a renowned scientist and have been there working as a, we can say, scientist in the field of biochemistry and science, working for many years in the Alakar Institute located at Pune, and dedicated his complete life to promote scientific thinking among the student and the society. Even though in the morning we had some of the discussions related to that, he is very much eager to have such sort of the talks, interaction with the students, that will come to know in few time, you can say, within this particular lecture as such. Along with this, he strongly believes that science should not remain confined to the laboratories, but should reach to common people. And that's why he conducts the lectures. So as I came to know that various institute for which he goes for the lecture, it's not just for the money, he doesn't want, but he wants to just promote the or inculcate the science as one of the important subject there in the students as such. So this is one of the fact that why he wants to say that he is confined. You don't want the students to be there as such, but confined to the laboratory and should reach to the common people. So he conducts the lectures, he writes or he has writing and public outreach program that inspires thousands of students as well as the scientists which are there all over the world as well as in India as such. Apart from this, his work reminds us that true education is not only about gaining the knowledge but also about developing the ability to think logically, and obviously the question around us worldwide that we have. So we get the solution from this. So I am 100% sure that today's talk, whatever he is going to deliver, will be related to most of us or is related to us, and that is going to help us each related to research as well as is going to help us in each and every part of our scientific knowledge as such. So let's take inspiration from him, from his dedication, as well as continue this work to promote the science and rational thinking in the society. So once again, I thank the sir on the behalf of the BPHC Society, from the chairman sir, from the secretary, as well as our principal sir, and from all of us. Sir, thank you for accepting our invitation, and thank you everything. I will say that as you are going to be here, and our students will be enlightened for the knowledge that we are going to have or we are going to deliver as such. So with these words, I once again thank you, sir. Thank you.
you, sir. I would like to take this opportunity to introduce our guest speaker for today. Our guest of speaker for today is Professor Dr. Surendra Ghaskadbi, a distinguished alumnus of our college. Dr. Ghaskadbi has completed his MSc in Zoology from Ahmednagar College in 1978, followed by MPhil and PhD from Savitribai Phule, Pune University. He is currently serving as Professor of Practice at the D.Y. Patil Biotechnology and Bioinformatics Institute. However, a major span of his illustrious career has been at the Agarkar Research Institute, commonly known as ARI Pune, an autonomous institute under the Department of Science and Technology, where he has been leading the developmental biology group since 1985. Dr. Ghaskadbi's pioneering research focuses on cell signaling during pattern formation, evolution of developmental mechanisms, and stem cell biology. His work employs a fascinating range of model systems, including chick, frog embryos, sponges, hydra, and human mesenchymal and embryonic stem cells. Remarkably, over the past 2.5 decades, he has introduced hydra as a model organism in teaching and research across India. He has played a prominent role in several scientific organizations. To name a few, he is a treasurer of Maharashtra Association for Contribution of Science. He is a past president of both Indian Society for Developmental Biologists and the Indian Society for Cell Biology. He is an elected fellow of National Academy of Sciences, India, and the Maharashtra Academy of Sciences. His research has received wide recognition and funding from major agencies such as DST, DBT, CSIR, DAE, and Indo-Japan collaborative grants, mobilizing over 4 crore rupees for developmental biology research. He has published extensively in reputed international journals, and his collaborations span both national and global scientific communities. Beyond his scholarly contribution, Dr. Ghaskadbi is a passionate science communicator who frequently engages with students through lectures, workshops, and outreach programs across India. Above all, Dr. Ghaskadbi remains a proud well-wisher of Ahmednagar College, his alma mater. We feel truly privileged to welcome him back to his parent department, the Department of Zoology. We welcome you, sir. Thank you.
Form and shape develops in animals. There are many similarities between plants and animals during their development, but obviously animals are very, very different from plants. So today we will stick more to the plants. And I am going to touch upon what kind of genes regulate development, what kind of cell signals are necessary, and how stem cells became such important part of not only biology per se, but the entire natural sciences and medicine. So I'll touch upon that also. As I have given in the abstract, I don't know how many of you have read it. Usually we don't read abstracts, I know that. But what I'm going to do today is use 2 stories of 2 Nobel Prizes that were given in medicine and physiology, and both are related to development. So I'll use that work to explain some basic concepts in developmental biology and where it stands today, and how it is important for all of us, and how it is also important for the general public to know as to how animals develop. Let me explain that we study animals for 2 basic reasons. One is natural curiosity, how a bat develops wings during development in spite of being a mammal, for example. Or, we are interested in how different animals develop. But there is another side to it, is that this knowledge from animals helps us understand human development. You all are aware that we cannot use human embryos for ethical, legal, moral reasons. And therefore we have to use model systems or research organisms to gain insights into what happens in human beings. And therefore we have to use animals. And so on one side it is scientific curiosity. So when you start doing work of that kind, you are not necessarily thinking of its applied value. Rarely we start scientific endeavors with something end product in mind. That's very rare. Many a times we carry out research out of scientific curiosity, but it turns out that it finally ends up into something very useful to human beings. And this very basic work that I'm going to tell you today about the 2 Nobel Prizes started purely as scientific curiosity and then ended up getting a Nobel Prize in medicine. So that is the journey. You start small and sometimes you are lucky enough to get something which is going to be very, very useful to your human race also. At the same time, you gain insights into the fundamental mechanisms of biology using any kind of animals. So we get different form and shapes in animals. And this is a random collection of pictures that I have collected from internet just so that I have something colorful. Of course, your dresses are more colorful than that. So there are several different animals. I have randomly chosen them. But I have also chosen 2 specimens from one species, Homo sapiens. Just to tell you that in spite of the fact that things have been conserved in evolution, as I will tell you, that developmental mechanisms have been conserved from the very, very simplest to the very complex organisms. But on the other hand, you have the same human genome, which can produce 2 so different individuals. So this is as per my age. You can choose any younger actor or whatever, Ishan Kishan or somebody from the current generation. But what I'm not going to touch upon how we become different in spite of having human genome. We don't have the time for that. But I just want you to remember that while I'm going to talk about conservation of mechanisms, there are also some fine tuning which makes each one of us in this room, in spite of having the same human genome, very different from each other. I don't see any identical twins, so we are definitely different. Okay? So let me just tell you certain terms that I'll be using. The study of developmental started as embryology. So people earlier used to look at only embryos. People like Aristotle. So you can imagine how old he was. Okay? They looked at embryos and they described how embryos develop. So Aristotle has some excellent pictures of chick embryo development, for example. Okay? And that time, chick embryos were easily available because everyone had a poultry. Okay? And frog embryos were also available because they are relatively larger in size. So all our earlier information, especially about the process called gastrulation, which makes mesoderm. We have 3 germ layers: ectoderm, mesoderm, endoderm. So mesoderm is induced due to gastrulation. So this was very descriptive to begin with. Okay, so people watched the development of embryos and described it. Later on, they started putting few things in the water where the embryos grew. For example, salt, or they changed the temperature, and they wanted to find out what is the effect of these changes in the environment on the development of embryos. So it became experimental embryology. You started to do experiments. So this was embryology to begin with. Later on, people realized that development is not restricted only to embryos. Development of an organism begins from gametogenesis, okay, right from spermatogenesis and oogenesis, fertilization, and then later on development till one undergoes aging and death. So to encompass all these areas, it became developmental biology. And in recent years, we know that developmental mechanisms have been conserved, so that came to be known as evolutionary developmental biology. Okay. You might come across this term sometime later in your life, or during NET exams and so on. So I will just explaining them. As an offshoot of embryology and developmental biology, we have certain very big branches now. For example, programmed cell death or apoptosis became a separate branch of biological endeavor only recently. But embryologists always knew that programmed cell death is necessary for making fingers, for example. Okay? If programmed cell death doesn't happen, these fingers will be attached to each other. And that is precisely what happens in case of ducks, where webs are formed instead of fingers. Okay? So programmed cell death or apoptosis, as its more recent term is an offshoot of development. It's very necessary for normal development. regenerative biology is also very important because we now want to see how regeneration happens naturally. For example, in organisms like Hydra, on which I work, although I'm not going to talk about it today, you can cut a Hydra into, let's say, 20, 30, 40 different pieces. Each one of them will become a new Hydra. So it is tremendous capacity of regeneration. You can cut a planarian, the flatworm, into 256 pieces, and each piece will become a new planarian. But as evolution progressed and we became more and more complex, we lost the capacity of regeneration. So, for example, we can regenerate only our liver to some extent. So, there are many different, theories as to why we have lost regeneration. But the idea is to learn more about regeneration and to explore the possibility whether ultimately human organs can be regenerated in vitro. And therefore, regenerative biology has become very, very important. Obviously, we cannot use human tissue, and therefore, once again, we have to go to animals. So, animals which show regeneration, we try to look at how they do it and to find out whether the tricks of these more simple organisms can be used to more complex organisms like the vertebrates, including humans. Okay, so that's the importance of regenerative biology. And stem cell biology, of course, you have heard about it. So always, during development, stem cells were always present, even in adult stem cells are present. But now it has become a separate field again, once again, because you want to use stem cells not only for treating diseases, but to also use them for organ regeneration, at least in vitro. So these are all the areas that encompass that, and many more that come under developmental biology. Depending on one's interest, one could pursue what one thinks. So how does early development take place? We are all aware of it, but just to refresh your memory, each one of us started our life as a single cell. Okay? So when the mother's oocyte is fertilized by father's sperm, we take birth as a single cell. And when we take birth, our size is as big as a full stop in any of the printed books. So that's small we wear. Now this single cell starts dividing, undergoing cleavage divisions. 2 cells, 4 cells, 8 cells, and so on. So it becomes a ball of cells, which is looks like somewhat like strawberry. Or better still, what you are more aware of is bundi color too, bundi color. Okay. So that's how we were all to begin with. But this ball of cells has to be now converted into a 3-dimensional organism. So for that, what happens during early development is that first axes are formed. So we have 3 axes, as you are all aware. Dorsal ventral is the first axis to be formed during development, then anterior posterior, and finally left right. So unless dorsal is formed properly, our vertebral column will not end up in the dorsal side. Unless anterior is formed properly, the brain will not end up here, right? We sometimes tease people that your brain is here, but don't. Brain is always here. If it was to be here, the person would not have survived. So once you come across, unfortunately, they have survived. So first the axes are formed. So it's like a architect's plan. You have a plan, and then you put things in proper position. So anterior is here, dorsal is here, heart loose on the left hand side in most people, so it becomes left right. That's the last axis to be formed. There are some people with heart on the right side, which is known as situs invertus, which is perfectly fine. But anterior, posterior, and dorsal ventral are very critical. In insects, I might mention that these axes are reversed. Those insects have a ventral nerve cord. We have a dorsal spinal cord. Okay. So once these axes are formed, different organs and systems are formed. Then beginning of this development of form and shape is known as morphogenesis. Morphology is study of form and shape. Morphogenesis is development of form and shape. And finally, you have perfect organs to be placed in different. So to begin with, you have a wall of cells where the cells are very similar to each other. And then as development progresses, cells start becoming different from each other, which I am going to discuss in the second part. So that what we call that as cell differentiation. Why cells have to become different? Because we are multicellular, and therefore we have division of labor amongst different cells. So some cells will become neurons, some will become cardiomyocytes, go to the heart. Only a small number of subset of cells in the pancreas will become islets of Langerhans, which will produce insulin. But please remember, and I'll repeat this again, that in spite of having so many different kinds of cells, and we have about 200 different kinds of cells, each cell in our body, even today, has the entire genome. So insulin gene is present here in my skin, but my skin never makes insulin. And why that happens, we will also discuss. So I have tried to give you a gist of what generally developmental biologists who study normal development do. And anything going wrong in this normal development produces abnormal pictures. So these are developmental abnormalities. Developmental abnormalities can be induced either due to hereditary reasons or due to environmental reasons. But all developmental abnormalities which occur at birth are known as congenital abnormalities. Congenital has nothing to do with genetics. Congenital abnormalities can either be because of hereditary or also because of environment. Environment means, suppose the mother who is going to give birth is an alcoholic, the fetus will have a smaller brain. That's also congenital abnormality. So by studying normal development or by studying abnormal development, you gain a lot of insight into normal development and also understand how genetic diseases are produced in early development. Okay. So why do we use certain model systems or research organisms? That is because we cannot study each and every one. It's not physically possible. Sometimes it's not logistically possible. So what scientists have done over the centuries is that they have chosen some representative organisms which help them in answering the kind of questions they ask. So I'll just briefly explain this slide. So there are some very popular model systems listed here. The one here is a worm called Sinorhabditis elegans. Okay? It's a tiny worm, microscopic worm. They develop in petri plates. They feed on bacteria. But the beauty of this system is that a single worm is made up of only about 1,000 cells, 1,000 cells. We are made up of trillions of cells and probably equal or many times more bacteria also. But these are made up of about 1,000 cells. So actually, if you can label one of these two cells, this is a 2-cell embryo. If you label the cell on the left, all the cells arising out of that labeled cell in the adult worm, you can actually see under a microscope because we have fluorescent dyes now. So you can follow which cell is giving rise to which cell in the early embryo is giving rise to which cell in the adult organism. So you can follow the trajectory of a stem cell to its differentiated state. So people who want to study those kinds of things will use Sinorhabditis, for example. Drosophila melanogaster, the fruit fly that you find on rotten bananas, has been one of the most useful or probably the most useful model system in genetics and development, and we'll discuss more about it. The main reason behind is that you can induce mutations here. So we have a number of genes. Suppose we have 25,000 genes. Now, if each gene is working properly, it is very difficult to understand the function of those genes. But if you can mutate one gene at a time and see the effect of that experiment, then you can understand what the function of the gene is. Do you follow me? Suppose there is a gene important in a certain phenomenon, certain mechanism or biological process. If you mutate that gene, mutation means you change the gene in such a manner that the protein that is produced is either defective or there is no protein produced, and then some phenotype, some morphological feature will appear. So if you damage a gene in Drosophila, and if the Drosophila is born without eye, then you know that the gene is important for eye formation. They call it eyeless because in genetics, many times genes are named after the phenotype they produce on mutation. So eyeless is actually necessary for eye. Wingless is necessary for wing. So if you produce mutations in one gene at a time, then you can understand function of many genes. And that is what the first Nobel Prize story that I'm going to tell about, those people have done. So Drosophila has been very, very useful. Then, as I mentioned earlier, frog embryos and chicken embryos, the one in E here, have been very useful to understand basic morphological processes, morphogenetic processes like gastrulation, because they were readily available. Another vertebrate model, these are both invertebrates. Another vertebrate model which has become very popular now is the zebrafish. Zebrafish embryos have become very popular because unlike the frog and the chicken embryos, these are quite transparent. Okay? So you can actually watch how development takes place in live embryos. Secondly, zebrafish has a tremendous capacity of regeneration. If you damage 70% of the zebrafish heart, even in the adult, the heart completely regenerates, which is a very rare case of organ regeneration in a vertebrate. So people would like to understand how these complex organs like heart are regenerated by zebrafish. And like in case of Drosophila, and unlike in case of frog and chick, you can induce mutations in zebrafish almost at will. So you have mutant lines. Suppose there is a line which carries a mutation for defective swimming, for example. So you can have a lot of such mutant lines in Drosophila. Like in Drosophila you can have in zebrafish, which is not possible in other vertebrate embryos, and that's why zebrafish is probably the most popular model system, vertebrate model system right now. And of course you have the mouse or the rat embryo, because you ultimately want to extrapolate your work to mammals also. So, and there are simpler model systems. Simpler in the sense, sponge for example, is a relatively very small, not the Rakhi sponge, this is the live sperm I'm talking about. So if you disaggregate, if you separate the cells of sponge, they come together and they form a new sponge. So one of the experiments that was done almost 125 years back is that a scientist, Wilson, brought a green sponge in the lab, disaggregated the cells, and then after a few days, the cells came together and they form a functional sponge. Next time he brought a red sponge. So, I'm talking about 1905, 1907. There were no markers then like GFP. So natural marker was, one was green color, one was red color. When he mixed the cells together of the two sponges, can you imagine what must have happened? This just to find out whether you are awake. Suppose you mix red and green sponge cells together. What will happen after, let's say, 5 days? Anybody? You are not in a class, you are allowed to talk. So what we found is that the green cells become separated and the red cells become separated. They sort themselves out. This is known as sorting out. So the conclusion of the experiment is that there is something in the green cells and in the red cells which they recognize self and non-self, which is the basis of immunology today, isn't it? Because they know what is self and what is non-self. Why do our liver cells and pancreatic cells don't mix up? Because they have different cell surface molecules on their cell surface. So this sorting out mechanism was first discovered in a simple organism like sponges. Hydra is another one. I told you that it has a tremendous capacity of regeneration. It's an evolutionary, very ancient organism, believed to have evolved 600 million years back. The first organism in evolution with an organized nervous system. Sponges do not have an organized nervous system. And there is also one which is not an animal, it's a mixed amoeba called Dictyostadium, which shows alternate between unicellular amoebae and multicellular stages. So you can study how multicellularity and unicellularity get intermixed with one another or get changed into one another. So these are also some model systems. This is not an extensive list. Depending on what kind of information you seek, what kind of question you are asking, you have to use either one, two, or many model systems to answer that question. And it will also depend on the facilities you have, what kind of lab you have, whether you have, for example, keeping mammalian embryos needs a lot of money because you need sterile culture. So there are many different criteria that one uses to finally hit upon a model system. Okay, now do you have any questions so far? No? Okay. Now let me go to the first novel prize that I am going to talk about. This was awarded to these three people: Edward Louis, Christian Naslin Wollard, and Eric Wishaus. Okay, their work was basically on Drosophila. What they discovered that how early embryonic development is controlled at the genetic level. Okay, they worked on Drosophila, and Edward Louis, after getting his Nobel Prize, clearly said that he found it very difficult to get funding, money for his work, because he was working on some fly that is found on rotten bananas. But ultimately, that got him a Nobel Prize in medicine in 1995. So he published his work in a journal called Nature in 1978, a very complex paper. So these three people, Naslin Wollard and Wishaus worked together, okay? And Edward Louis worked separately, a senior guy. So let me explain their work. They worked on Drosophila, as I said. So this is Drosophila fly. I don't know how many of you have seen Drosophila. Many of you must have seen. The red eyes are very, very clearly visible. So this is the entire body of Drosophila. This is the dorsal side of Drosophila. This is the ventral side. Okay? This is anterior, this is posterior. The entire body of Drosophila is made up of 14 segments, 1-4. So head is formed of 3 segments. Thorax, the middle part, is formed of another 3 segments. And the abdomen is formed of 8 segments. So animal bodies are segmented. Our body is also segmented, which is very clear from our ribs or the vertebrae, not the 6 packs. That's how it's happening. So, three head segments, 3 thoracic segments, and 8 abdominal segments. 14 segments make the entire fly. You will notice that the head has 3 main structures. It has a pair of eyes. You can see one eye because this is a lateral view. There is a pair of tentacles, the tiny tentacles here, which are sensory organs. And there are mouth parts for the fly to feed. These are the 3 major parts of the head. In the thoracic segment, all the 3 thoracic segments have 1 pair of legs on the ventral side. Okay, so it has 6 legs. All insects have 6 legs. That's why they are called as hexapoda. Ticks and mites are not insects because they have 8 legs. Okay, but otherwise all insects have 6 legs. So every thoracic segment develops 1 pair of legs on the ventral side. On the dorsal side, the second thoracic segment has 1 pair of wings. So unlike butterflies, Drosophila has only 2 wings. Okay, and that develops from the second thoracic segment. And the third thoracic segment has a pair of balancers called as arteries. Okay, but there is no such structure on the dorsal side of the first thoracic segment. And all the other organs are in the abdomen. Now the question the scientist asked, in a very simple terms that is understandable to all of you, is why eyes develop here? Or why wings develop only on the second thoracic segment, but not on the first and third? Or the halters develop only in the third thoracic segment, not elsewhere. And also the legs develop only on the thoracic segment on the ventral side, but there are no legs in the abdomen. While in other animals like millipedes and centipedes, you see legs everywhere. So what regulates this? What decides this? The ultimate aim was to understand why we have 2 ears, 1 nose, 2 eyes, 2 hands, no tail or horns, right? So what is the mechanism that guides this? Now remember, if anything goes wrong in normal development, it will give rise to an abnormal fly. And I'll come to those examples. So these 3 people set about, not only 3, there are many other people who also did the study. Please remember that any discovery in science or any Nobel Prize in science is not because of just those individuals, but we stand on the shoulders of giants. Okay, whatever previous information is available, we build on that. But some people who do the exactly path-breaking discoveries only get the Nobel Prize. So to understand how this happens, I must introduce to you some with some simple facts of Drosophila development. So this is the Drosophila egg, okay, which is fertilized. And this egg has one nucleus. You know, one cell has one nucleus in our case. But in case of Drosophila, in early development, Drosophila nucleus undergoes division, but the cytoplasm doesn't undergo division. So what you get is a single cell with many nuclei. You understand this? So this is known as syncytial embryo. Okay? So until this time, stage 9, you have hundreds of nuclei in the embryo, but there are no cell membranes. It's a single cell. So you can imagine that this entire cell has cytoplasm which can freely diffuse. There are no cell membranes in between. There are no obstacles. There are no speed breakers. But at a certain stage, the nuclei start developing membranes around them, and it becomes, from a syncytial embryo, it becomes a cellular embryo. So now every cell has just one nucleus. The beauty of this system is that up till this stage time, the flow of molecules from, let's say, from the top to the bottom or from the left to the right is very easy because it has to only diffuse. There are no membranes in between. After this stage, of course, it becomes slightly more difficult to transfer signals from one place to another. When I say molecules, they are essentially mostly protein molecules and they work as signals. So what these Nobel laureates did, this is a summary slide I'll explain to you, is that they found out groups of genes which actually regulate Drosophila development. So initially, they showed that in the embryo itself, before it is fertilized, there is unequal distribution of molecules. For example, you can see that the red molecule is more on the left hand side and the green molecule is more on the right hand side. So this is provided by the mother herself and therefore it is known as maternal effect. Okay? This has nothing to do with the sperm or fertilization. It's already there in the embryo. So if you take a Drosophila embryo, things are not evenly distributed in the cytoplasm. They are unequally distributed. There is always a polarity. For example, red is more on one side, blue is more on the other side. As a consequence of this unequal distribution of molecules, certain genes are activated in the embryo later on through another protein called hunchback. Now, some of these names can be very confusing, so you need not remember the name. But essentially, hunchback means if this gene is mutated and if hunchback protein is not produced, the Drosophila becomes hunchback. Right? That is the phenotype there. As a consequence of hunchback, you get expression of genes which are known as gap genes. They are known as gap genes because if these genes are mutated, there are gaps in embryos. So for example, if this yellow gene is mutated, that part of the embryo will not grow. Right? So they are known as gap genes. Why gap genes are important? Because they roughly divide the embryo into discrete parts. See, suddenly to change an embryo into an entire final structure is difficult. You know the building is built brick by brick according to the plan. So likewise, embryo is also built like that, brick by brick. As a consequence of the expression of gap genes, you get pair rule genes. I told you that Drosophila has 14 segments, 1-4. You will see that there are pair genes are 7 in number. So these 7 decide how 14 segments will be formed. Let's not go into mechanism right now because it's very complex. You need not know at this stage. But I can answer any questions later on. These pair rule genes then activate 2 types of genes. One is segment polarity gene. So suppose there is a segment, suppose I am a Drosophila embryo, there is a segment here. This segment, how will it know which is its anterior part and posterior end? So this is anterior, this is posterior. These genes decide the polarity of individual segments. And finally, the last group of genes, which are known as homeotic genes, decide which structure will develop on which segment. For example, hands will develop on this segment, which is decided by the homeotic gene. And these were discovered by Ed Lewis, the first Nobel laureate that I showed. All the remaining genes were discovered by Naslin Vollard and Eric Bischoff. These are artificial colors given to those gene expressions. So you can actually visualize these now under a microscope and provide artificial colors. But under a confocal microscope, you can also give various different fluorescent dyes and actually visualize them. So you can see these are the gap genes here. This is a big gap. If this gene is mutated, the embryo will be short by that piece. These are pair rule genes, and this is segment polarity genes. So how does this happen? The maternal effect gene, how do they function? The first group. This is an oocyte, yellow oocyte, developing inside a Drosophila. And it is surrounded by some nurse cells. Nurse cells means they are essentially nursing the developing oocyte. The nurse cells deposit molecules in the oocyte in such a manner that the red molecule, which the names I will come to, is deposited on one side here, while the blue molecule is deposited on the other side. So this oocyte is still growing, it's about to mature, but already its cytoplasm has different molecules in different places. So this is known as bicoid, this is known as nanos. Red is bicoid, blue is nanos. So what happens? What is the consequence of this unequal distribution of molecules? So please see here, this is the red molecule deposited in the oocyte. It is deposited in the form of messenger RNA. It has not yet formed the protein. Okay? When the mother's bicoid gene is expressed, it forms the transcript, which we call as messenger RNA. This messenger RNA is deposited on one side, and as fertilization takes place, this bicoid messenger RNA gets translated into bicoid protein. Transcription, translation, right? So this bicoid protein gets synthesized on the left hand side in this figure, and because it's a single cytoplasm, there are no cell membranes, the bicoid protein diffuses from the left to the right because of simple laws of diffusion in chemistry, right? As a consequence, you find more molecule on the left and the concentration goes on falling, and ultimately you have no bicoid molecule on the right. Is it easy to understand this much? It so happens that wherever bicoid is present in the highest quantity becomes the Drosophila anterior part, the head of Drosophila. So what people do in early embryos is they take out the exoskeleton. So insects have exoskeleton, skeleton outside the body, and they make a preparation which you can observe under microscope. This is the actually external phenotype or structures that you see. So this is a normal animal, and this is a bicoid mutant. It has no bicoid protein. As a consequence of that, it doesn't form head and thorax. In normal, you have head, thorax, abdomen, the three parts. And of course, there are two extremities called acron and telson, which we are not considering right now. Head, thorax, abdomen. But if you mutate bicoid gene in Drosophila, you get only abdomen. There is no head and thorax. It means that bicoid is the protein that specifies head and thorax structures. So if bicoid is absent, head and thorax do not form. That is the conclusion from these initial mutation studies. Likewise, if you do the same experiment with nanos, mutate the nanos gene, which was on the right hand side, the blue gene, what you see is that head and thorax are formed normally, but abdomen is not formed. So the conclusion is that bicoid is necessary for formation of the anterior structures, and nanos is necessary for the formation of the posterior structure. Okay? And the two extremities are specified by other genes like torso. We will not consider that, it's too complex. So what we have now, two molecules, one is bicoid for anterior structures, one is nanos for posterior structures. This is from mutational analysis. So what you do is, you expose Drosophila flies to chemical and physical mutating agent and then look for mutants in the next generation. Okay, Drosophila breed very fast. So in the lab, you can keep several generations of Drosophila. See, if you want to study mutation, then you must have an organism which reproduces fast. Generation time is less. Otherwise, if you want to do PhD in elephant reproduction, maybe they will reproduce only after 18-20 months. So it takes time. I'm just giving a exaggerated example. Okay. So with Drosophila, you can study several multiple subsequent generations for mutation. Okay. So how can you prove this? In the lab, what scientists would do to prove that bicoid is important in the formation of anterior structures. And these experiments were done long back in the 80s. Therefore, they used autoradiography. Now we use only non-radioactive probes. But here what they have done is, they have used radioactivity to show the localization of the bicoid mRNA. So this is actually an embryo. Then at a later stage, this bicoid messenger RNA gets translated into proteins, as I mentioned earlier. But being a transcription factor, bicoid is a transcription factor, so it resides in the nucleus. You know, transcription factors work in the nucleus. So you can see bicoid present in individual nuclei, but you can also see the gradient of concentration. So bicoid is present somewhere up till only here. After that, it is not present at all. You can actually measure radioactivity by using equipment, and you can plot a graph. So in a normal embryo, if this is considered 100%, the bicoid concentration will go on falling as you go from left to right. But if you take a mutant and look for actual measurement of bicoid protein, you see that it is negligible. It is so low that these flies will never form anterior structures. And that's why you end up getting embryo with abdomen. Please remember, these will never turn into flies because these are lethal mutations. So you can see them only in the embryonic stages. So clearly, bicoid is necessary for formation of the anterior structures. How do you confirm it further? Because scientists, and especially when we published our papers, the referees, the reviewers are never happy with whatever data is there. They want more and more data to make sure. So how do you do these experiments? Typically, in embryology, such experiments are carried out. So you have this normal fly. You know, in genetics is known as wild type. Okay? So in a wild type fly embryo, you have bicoid mRNA on one side, and wherever bicoid is present, anterior will form. So you have a normal pattern. Acron, head, thorax, abdomen, tail. This is head to tail, wild type phenotype. Okay? But if you mutate bicoid, as I said, you will get only abdomen. So how do you do experiments? Well, I will take this fly, which has no bicoid, and inject bicoid messenger RNA from outside. We can do it because now recombinant DNA technology is fairly available to everybody. So you inject bicoid from outside in a mutant embryo, and you can see that its pattern is normalized. So bicoid is clearly necessary for anterior formation. You can go a step further because we are never satisfied, and we also want some spectacular figures in our papers. So what you do is instead of injecting bicoid in its normal place, you inject it in the middle. Now you end up getting head here, two thorax pieces here, and then abdomen. So making sure again and again that bicoid, wherever bicoid is expressed, cells around that will form the head or the anterior part. Or you can tell a normal fly, okay, and inject additional bicoid on the other end. So bicoid is already present here. You inject bicoid here, you get a two-headed fly. So you get two heads. Please remember again, these are lethal mutations because the cells are not sufficient to form any structure. Only available cells you are. How do you know this is head, thorax, and abdomen? Because we have molecular markers. There is no head per se, but we have genes expressed in head. They are seen here, and therefore you can actually see what kind of structures are going to be formed. So this is rather compelling data to prove that bicoid is responsible for anterior formation. So those two Nobel laureates, Naslin Holland and Eric Strauss, did some 8,000 such mutations of individual genes and hit upon many hundreds of them which are responsible for the regulation of different structures in early development. Okay. Ed Lewis did something which is entirely different. So far we have been talking about how anterior is formed, how head, thorax, abdomen is formed, how segments are made, how segments become anterior and posterior part, and so on. But another crucial question is why wings develop only on the second thoracic segment. So what Ed Lewis discovered, very dramatic discovery, is that there are groups of genes which are known as homeotic complexes, he called them. He found 8 genes in Drosophila which are present on a single chromosome and which specify different structures from the anterior to the posterior. Please remember that generally our genes are randomly distributed in our genome. It's not necessary that the gene for my eye formation, retina and cornea will be nearby. They can be different on different parts of DNA. However, in this case, Ed Lewis discovered that there is a single chromosome on which there are 8 genes. He grouped those genes under 2 different groups. One is known as antennapedia complex, the other is known as bithorax complex. And what he showed was, now you look at the color code, the gene here called labial, don't bother about names, specifies only this part here in the head. If you go to, let's say, this gene, abdominal A, it specifies only certain abdominal segments. So these corresponds to these. So actually from the anterior to posterior side, genes are arranged on the same side from 3' to 5' position on a chromosome, which sort of he discovered as to which genes are responsible for formation of which structure in the Drosophila. I will not go into technical details very much, but let me give you 2 or 3 examples. So just compare, this is the embryo, this is the adult. So you can see the color code. Let me give you just 2 or 3 examples of the homeo box genes. This is a normal Drosophila head under an optical microscope. So you have 2 red eyes, there are 2 antennae in the middle, and these are the mouth parts. If you look at this head, fix the head in some fixative and look at it under a scanning electron microscope, you will see these 2 beautiful eyes on either side. These are the 2 tentacles and this is the mouth parts. Now, once you discovered which gene is responsible for leg formation, what you can do is you can express that messenger RNA for leg forming gene at the place of the antenna in early embryo. And what you get is a Drosophila with legs on its head. So you can clearly see these are legs. These are once again very rare mutations. So please don't go outside and look for Drosophila walking on their head. But what it shows you, it's restricted only to structures. Okay. So what it means is that when you express that gene in this place, the cells surrounding it, okay, the original cells, instead of forming antenna, get a different instruction now, and instead of antenna, they form leg. So there are specific signals which are necessary. So these signals change, and as a result of that, legs are formed. Another example. I told you that Drosophila has only 1 pair of wings from the second thoracic segment and a small pair of halteres, the balancing organs. You can overexpress the wing forming gene in an early embryo and you can get a 4-wing fly. Okay? Or you can express the halter forming gene in both these segments and get a fly with only halteres. Now, this fly can fly but cannot balance itself, while this fly cannot fly, right? Because it has no wings. By simple manipulation of almost 1 gene, you can make the third thoracic segment develop a wing in place of halter, or a second thoracic segment to develop a halter in place of wing. So Ed Lewis discovered all these genes which are responsible for different structures of Drosophila. Somebody developed an eye on the leg of Drosophila. So eyeless gene was expressed on the leg. Those eyes were formed. Okay? This eye is not functional because there are no nerves there. But it has a perfect structure, as you can see. Red eye. Some other scientist, there is a lot of jealousy. He was not happy with that. He produced hundreds of eyes on the back of a Drosophila. Okay? So, back of a Drosophila full of eyes which were not functional, only structure. So, by changing the expression of genes, which we call in technical terms, we call it ectopic expression, not at its proper place, at a different place, ectopic. Then you can change structures. But what these abnormal embryos tell you is that what is the normal function of the gene. So wherever you put it, you will get an eye. Obviously, the gene is necessary for the development of the eye. I have another 10-15 minutes. But then why this tiny Drosophila work in Drosophila get a Nobel Prize in medicine? It turns out that after some years, people discovered that mammals have similar genes. The only difference is that in Drosophila you have only one set of such genes. In mammals we have 4 sets of genes. So not only in mouse, even we have these genes, which are known as OX genes. And these are the genes which decide which structure will develop along our anterior-posterior axis. So we can predict about the causes of developmental abnormalities, genetic abnormalities, based on this work, and that is the reason why the Drosophila work ultimately got a Nobel Prize in medicine or physiology. Okay. Why we have 4 clusters? Because the more genes you have, the more DNA you will have to put in the nucleus. The more DNA you have, more chances of environmental factors damaging the DNA. So, if you have more genes, that means they must have some evolutionary advantage, and that's why we have retained 4. And we now know that if A1 gets mutated, B1 can take over its function. So there is redundancy of function, which helps us develop normally. If there is a problem in one gene, similar gene will take over its function. So that is why this cluster has been duplicated twice in evolution. We have 4 clusters, and that's why we can manage, getting away with several abnormalities. So this is a comparison between Drosophila and vertebrate development. I just want you to look at the color code, see how much similar. We are not all that different from a fly. Okay, we may, like to listen to Carnatic music, but other than that, structurally we are just another animal species. Once again, a Drosophila embryo and a mouse embryo. So you can see the similarity here. We are very, very similar. In fact, in some cases, you can replace the gene. You can express the Drosophila gene in mouse and it will carry out a normal function. You can see that certain diseases give very similar pattern in, let's say, a rat and a human being. This is a gene called this is a disease called P-biotism. There are many symptoms, but one of the symptoms is lack of melanization. Okay, so you can see very similar spots, and that's why we use models, animals as models, because we cannot do these experiments in humans. So, in spite of having so much conservation of mechanism, we have similar genes as Drosophila uses. Then how come we have so much of biodiversity in spite of having similar genes? They should all produce a similar kind of animal. But simple example, as I mentioned earlier, simply because there is no cell death here, ducks can get a web, chicken will get fingers. They are very, very similar, both are birds. So their genes will be very, very similar. But still, by just changing where and when a gene is expressed, you can make different structures from the same set of genes. Another example. This is a cartoon of a vertebral column of a bird on the top and a mammal at the bottom. Okay? So this is a chick vertebral column, this is a mouse vertebral column. We know that we have only 7 neck vertebrae, whether we or giraffe have 7. Only the giraffe vertebrae are longer, but the number is always 7. While birds have more number of vertebrae because they have longer necks. I'm considering the body here. So what you see here is that these genes, the mammalian counterparts of the Drosophila genes, hawks, the hawk's 5-6 boundary here is shifted towards the anterior side here. Can you notice that? See, where hawk's 5 expression stops and 6 expression begins is shifted here. So hawk's 5 stops here and hawk's 6. As a result of that, only 7 vertebrae can be found in mammals because hawk's 5 is required for that vertebra formation. And these hawks 9 and 10 are somewhat similar. So thoracic vertebrae are less in birds because more neck vertebrae are there. While in case of us mammals, cervical vertebrae are only 7, thoracic vertebrae are more. But the genes are identical. See, almost the same genes. Just shifting their boundary makes a different structures. And that is why, on one hand, you have conservation of developmental mechanisms throughout all the animals, but by simply tinkering how much the gene expressed, where is this expressed, how much it is expressed, you can make either a Drosophila or a monkey or a giraffe or a human being. Okay, have you understood so far? Okay, so this is just Darwin is the one who proposed the theory of evolution, and this is a cartoon because they used to ridicule Darwin in this old cartoon of 1881 in Punch magazine. That cartoonist showed this worm itself getting evolved into Darwin. They were actually making fun of that. But we now know that we have a common ancestor. This is how Darwin looks, as you know, when he got recognition. This is how he looked when he went to work. So in science, you take a very long time to get recognition. And Nobel Prize, if you want to get, you need to remain alive. So it is better to maintain your health. Nobel Prizes are given only to people who are alive. See how much time it takes to. So grey hair, although not all grey hair are respectable, usually hair turn grey by the time you get recognition. Okay, now let me take another 10 minutes for the second story. We have time? Okay. Are you okay with continuing? So the second Nobel Prize was given in 2012 to two scientists, Sir John Gurdon and Professor Shinya Yamanaka. And this was given for their finding, see the last sentence, they discovered that mature specialized cells can be reprogrammed to become immature cells. So what I told you is that as development progresses, our cells differentiate. And suppose insulin gene is not required for in my skin, that gene will be switched off. We always believe that genes are switched off permanently as development progresses. But the finding of these two people showed us that genes can be still activated in differentiated cells. I'll quickly tell you what interesting experiments they did. So let me first quickly define stem cells. A stem cell is a cell which must have at least two characteristics. One is it should self-replicate, it should be able to divide, and it should be able to differentiate into 1, 2 or many different cell types. For example, neuron or erythrocyte, muscle, and so on. If it differentiates only in one cell type, it is known as unipotent. Or bipotent is it can differentiate into 2 types, or multipotent. The best example of this is our hematopoietic stem cells, the stem cells which are present in our bone marrow. They give rise to all the cells in our blood, but they are present in our bone marrow. The immature stem cells do not come into circulation. If they come into circulation, we call it leukemia. Only differentiated cells are found in the circulating blood. And also I would like you to notice that this cell can self-replicate as shown by the red arrow. But when they differentiate into different cell types, they don't directly come from this stage to this stage. They undergo different steps. Okay? So that's necessary because from a single type of cell you have to produce multiple kinds of cells. So there are different steps. As development, this is an example of a frog embryo. As development progresses, you have initially only ectoderm, mesoderm, endoderm, and as we go developing, we get more and more cell types. I have already told you, so I will not repeat that. That is known as cell differentiation, and this cell differentiation occurs because of differential gene expression. Although all cells in our body have all the genes which are not required for a cell's function are switched off. For example, in cardiomyocytes, only the muscle cells will be more active. Insulin gene, for example, is inactive. Or keratin gene, which is important for skin, is inactive in heart, right? And this is because there are certain housekeeping genes. Housekeeping means they are required for reproduction and metabolism of all cells. They are active in all the cells in our body. Otherwise, the cell will die. But only specialized genes are active in specialized. For example, there are genes required for the production of milk in a lactating mother. Otherwise, they are switched off, right? But they are present. So what Gardner did was very interesting. He took a frog and took its egg and removed this nucleus from this egg by irradiation. To understand the experiment, very simple. Take eggs, destroy the nucleus. Then he took another frog and took a differentiated cell from the skin. So in a differentiated skin cell, most of the genes not required for the function of the skin are switched off. Keratin genes will be active. What he did was, he took nucleus from this cell and put this nucleus into this cytoplasm from which the original nucleus has been removed. But this is an oocyte. And he got a clone of the frog. Okay, so this is the result. What it means is, this nucleus, which had switched off most of its genes, could switch on its genes when put into a new environment and can actually produce a clone. When you produce a clone, that means every gene is active. Otherwise, you cannot produce an organism. Right? Just to make sure that the nucleus was responsible, we took a wild type xenobus embryo and introduced differentiated nucleus from an albino strain. So these have no melanocytes, no melanin, so they are albino. And when you take nucleus from this, all the clones that are produced are albinos. This experiment he did was in 1956-1957, the year I was born. Ancient, your parents probably were also not born. Okay? These results were so spectacular that nobody would accept that. So what John Gurdon did was, he kept these frogs in Cambridge, UK, where he worked, and went to United States of America for a postdoctoral fellowship for 2 years. When he came back in 1960, later part of 1960, he saw that all his clones were not only alive, but they were reproducing. So they were all functional clones, right? So he published his work in 1962, for which he got a Nobel Prize in 2012. I told you to remain healthy. He got it after 50 years. Professor John Gurdon died only 3 months back at the age of 92, 93. And based on that, Dolly was cloned. You know about Dolly, but you don't know about Gurdon because those were the real experiments which we have studied as textbook experiments. So what Ian Wilmot and his group did was, they took nucleus from a mammary gland cell. I told you mammary gland cells are terminally differentiated, right? Do you understand? Do you agree with that? And then he took the nucleus and he took an egg from a different genetic background. These are different strains of sheep. Removed the nucleus and put the nucleus from the mammary gland from this into this enucleated egg cell. They started multiplying as an embryo. This was put into a third type of sheep, surrogate mother, right? Because he did not want any confusion. This has a different genetic makeup, this has a different genetic makeup, and this has still different genetic makeup. And finally, this Scottish blackfish sheep gave rise to Dolly, which is of course like this, clone of this, because the nucleus came from here, so the genes came from here. So that was the basis of Dolly. Ian Wilmot is also no more. Now, of course, there are shops in China, and I'm sure at different places. This is an old news item from Times of India. You can make a clone of your dog for $53,000 and cat for $35,000. Now, I'm sure it is much cheaper. So now there are shops who do cloning, pet cloning. This is at least 7-8 years old news item. So Gurdon's work was responsible for animal cloning. They also cloned long-tail macaque. Zhongzhang and Hui, I cannot make out who is who. They are identical twins, clones actually. So you can produce monkeys. I'm sure some rogue scientists may be trying to clone humans also. What I'm trying to tell you is that in principle, the technique, though extremely difficult, in case of Dolly, for example, hundreds of embryos were transferred, but only one Dolly was born. Although they are very difficult, in principle, it is possible to fool a differentiated nucleus by putting it into a oocyte cytoplasm to believe that it has to express all its genes and make a clone out of it. Okay? The other Nobel laureate was Shinya Yamanaka, who did entirely different work but with the same principle. He took differentiated cell from the body. I told you differentiated cells switch off most of insulin. But he did certain kind of reprogramming by which these differentiated cells were converted into stem cells, pluripotent stem cells. Since they were induced experimentally to form pluripotent cells, they are always written like this. I for induced, E and S capital. And these induced pluripotent stem cells now can be differentiated into muscles, neurons, or erythrocytes or whatever. The beauty of this work is the following. You can take a sickle cell anemic mouse. You know, in case of sickle cell, there is a point mutation in the hemoglobin molecules, as a consequence of which cells become sickle cell. They are unable to carry oxygen to the full extent. That's why the patient gets anemia. So, you take a sickle cell anemic mouse and collect skin cells from this mouse. They are converted into induced pluripotent stem cells by introducing these 4 factors, which are known as Yamanaka factors. He used hundreds and thousands of combinations of these factors and hit upon these 4. OCT4, SOX2, KLF, and CMYC. These are known as Yamanaka factors. By doing this, these skin cells were converted into stem cells. And because anemia is dependent on a point mutation, he corrected the point mutation in these cells, and after the correction of the mutation, the cells were grown in larger numbers and put them back in the patient, and the patient recovered from anemia. This kind of experiments have not been done in human, but trials are going on in which, for example, I am a diabetic. Till yesterday I was not. But what you do is, suppose I cannot produce enough insulin. Instead of taking insulin from outside, what people are trying to do is to take my skin cells, convert them into induced pluripotent stem cells, and then convert them into islets of Langerhans. Put them back in a pouch near my kidney, usually they do it that way. That will start secreting insulin and my diabetes will be cured. The beauty of this system is the cells are mine and therefore no question of rejection. So these kinds of trials are going on, which are based on Yamanaka's work. So Gurdon and Yamanaka showed that you can activate suppressed genes in differentiated cells, and that furthered our understanding not only of normal development but also of stem cell biology and regenerative biology. So just I want to introduce one concept here before I show you some pictures of these people. In normal development, a cell, so this is the early development, as it goes down, it starts differentiating. That is the normal trajectory of cells. So from a stem cell, you get a differentiated cell. Sometimes, if you use Yamanaka-like experiments, you can reprogram a cell. So cell which is already differentiated, you can take it back. This was not known till Yamanaka did his experiments. We only knew this trajectory. But there are also cases in normal development where some cells automatically, we don't understand the exact mechanism, become from one differentiated cell type to different differentiated cell type. They don't become stem cell, but they are changed. That is known as transdifferentiation. So, Conrad Waddington, a famous embryologist who worked on duck and chick embryos, proposed a term called epigenetics. You must have been aware of that. And according to Waddington's epigenetic landscape, what you have is you have the pluripotent stem cells in development, and as development progresses, through steps they become differentiated, right? Some of them can be transdifferentiated by mechanisms that are still not fully understood. However, Yamanaka also told us that these cells from a differentiated state can go back to pluripotency. Okay. Now we have a different branch as epigenetics, but when Waddington proposed that theory, that term epigenetics, he was talking about the connection between genetics and developmental biology, which was not understood then. Just to show you, when Waddington visited in the late 1950s the zoology department of Pune University, where he met Professor Leela Molerkar, who did her PhD with Waddington in 1956 and started embryology. And this lady then, I joined her for my PhD. So although Waddington and Molerkar may not agree because they are no more, I consider myself as Waddington's academic grandson. Just to come back to Gurdon and Yamanaka, Gurdon was born in 1933. He passed away this year. Okay, so until very recently also came to India. I'll show you one or two photographs. Yamanaka was born in 1962 when Gurdon published his work. But both of them got Nobel Prize together. Because Yamanaka published his work in 2006, but got his Nobel Prize within 6 years, which is a very rare occurrence. Within 6 years he got his Nobel Prize, while Gurdon got it after 50 years. So John Gurdon until recently worked in his lab. Until his last day, he was working in an institute called John Gurdon Institute of Developmental Biology. There is an institute after his name in Cambridge, UK. He is a normal person. People think that scientists are very boring, not at all. So he is with his students. Here he is showing how a frog can leap and so on. He was in Pune University in 1993, and this is me. Just to impress you, I will put this photograph. This is another developmental biologist. So way back in 1993, he had come to Pune University. Later on also, he came to India many times. I was fortunate enough to interact with many of these developmental biologists. There was a meeting in 1998 in Nainital. Why I'm showing you this? It's very important to meet such people. It's not, you cannot sit in a small room and you cannot, you have to exchange ideas with people. You have to learn from such people. So you can see somewhere here, Gurdon is here. There are many scientists here. Of course, I am here. Then once again in Sikkim, he had come. You can see him here. So scientists have a lot of perks. We have meetings in excellent places, So it's not at all boring. When we go out, we really, what do you call it, chill. Here he came, he actually examined the thesis of my student in 2008. He came to Pune University. So Smruti, she worked on stem cell biology, so he was one of the examiners. And finally, I'll leave you with a slide of Gurdon's report by his science teacher when Gurdon was in school. Okay, this is from 1949. It has been a disastrous half. His work has been far from satisfactory. His prepared stuff has been badly learned, and several of his test pieces have been torn over. I remember my journals in zoology department. One of such pieces of prepared work scored 2 marks out of 50. I think we are all with John. His other work has been equally bad, and several times he has been in trouble, and this is important, because he will not listen, but will insist on doing his work in his own way. I believe he has ideas about becoming a scientist. On his present showing, this is quite ridiculous, says his science teacher. If he can't learn simple biological facts, he would have no chance of doing the work of a specialist. And it would be sheer waste of time both on his part and of those who have to teach him. So my take-home message is that, with apology to the first bench, don't take your teachers seriously. Any questions? They're all tired.
Hello.
Huh?
I knew it would be a very wonderful lecture. I have been listening to you since 2005, since I was a student.
And you still attended again?
Yes.
Oh my God.
So, you said that there is no permanence or no switch off for genes. So, you talked
Naturally, there is switch off, but experimentally you can switch
in
on.
principle, you said.
Yes.
So, you talked about reprogramming. So, nowadays we are talking about the reversing the aging process to remain healthy and all. So, how do we see this reprogramming in reversing the aging process?
So, the term aging we have been using rather loosely. With the kind of advertisements that you see for remaining young, or Deepak Chopra's lectures and all. He's in Epstein files, but he used to talk a lot. So, as far as science is concerned, we have not hit upon a combination yet, which will help us reduce aging to some extent. Because it's a very multi-disciplinary field. In the sense, you need to have a very good DNA repair system. You need to have a very good metabolism. So it's very complex. When you talk about this kind of work, there is often correlation between one or two or a small group of genes with the biological phenomenon. Aging is too complex. I think out of our 20,000 odd genes, probably everything is involved in aging. So we have not come to that stage. But what we have understood from animals is that at least you can use these tricks of animals which do not age. For example, Hydra doesn't age. It's supposed to be potentially immortal. If you can use these tricks, at least in vitro, to grow stem cells and use them for organ regeneration in vitro. To that stage, we are fairly confident. But aging, of course, is an issue. With all this population, I don't know why people want to stay. Where will we live?
Another question I have is nerve cells.
Yes.
Terminally differentiated. So they are arrested in G0 phase of cell cycle. So again, how do we look at?
So now there are some reports that nerve cells can also regenerate. There are few reports, but the solution to this is to generate new nerve cells from pluripotent stem cells. But once again, all said and done, putting a stem cell in a human patient is a very risky affair, because you do not know how it will behave. A stem cell will divide, and we are not sure whether it will stop at the right time. Otherwise, you end up getting tumors. So stem cell therapy is difficult because we have no control over how it will behave inside the body. That's the problem with the stem cell biology right now. So in principle, we have the basic knowledge. But as you appreciate, and as I hope all the students appreciate, that we actually know very little. We don't know how hormones control our moods, and it's very complex. So I don't think we have reached that stage. Although we talk a lot, I think our ignorance is much more than what we know. So we need more and more younger people to come into science because there is so much more to learn, right? And it's not only in the genes, but also the environment. So identical twins growing in two different countries behave differently. So it is nature plus nurture. Or it's no more on or nurture, it's nature and nurture. So it's very complex. It's very difficult to answer that question. But there are exciting leads. Let's put it that way.
So continuing to the same topic. So do we really preserve the cord blood cells then?
So this probably still fashionable. See, cord blood cells, if preserved properly, maintained properly over a long period of time, may become useful for certain diseases in siblings or the same person. That's possible, in principle. Because we have shown that there is empirical data, there is clear-cut scientific data to prove that. The problem is logistics. You keep it in liquid nitrogen. Now, to keep such cells in liquid nitrogen for, let's say, 18, 20, 22 years, are the stem cell banks doing it properly? Okay. Do we have that kind of money? And whether after 20 years down the line, will the stem cell be really revived so that they can actually differentiate into cells that are required? These are questions which I am not fully comfortable with. But science-wise it has been proved. But I donate my child's cord blood cells today, which is because they are going to go waste. So that makes sense because they are going to be thrown away. But whether they will be maintained properly for 20-25 years and whether they will revive into normal cells or whether some abnormalities will be induced in them, we don't know yet. And I don't know whether there will be sufficient liquid nitrogen supply. You need generator even for such a talk. So imagine 20 years you have to keep them in a condition which is favorable. Practically it sounds a bit difficult. So I'm not a big fan. People have personally asked me, a couple of them, although I'm not a clinician, I have advised them against it. Maybe after 20 years, some of these diseases may find better cures. We don't know. But these are questions that we can't answer. Yes.
So, sir, I had a question.
Yeah.
That do you think religious ethical objection to stem cell research can be justified? If why?
What are the objections?
Like some religions oppose the stem cell research because they go against the creation or they go against God. So how can it be justified?
So very, I'm not really very competent to answer this question because I'm an atheist. I don't believe in God. I believe only in scientific facts and I believe in evolution. So question of creationism and somebody creating an organism doesn't appeal to me at all. I'm an atheist. I don't believe in God.
So when we do ectopic transplantation, the cells need to be competent. So how do they overcome this and grow?
Okay. So the question is, when we do this transfer of cells from one place to another, how do they remain competent? So in early embryo, when these transplantations are done, there is no immune rejection developed. So there is no rejections, but that develops later in life. So when we do organ transplantation, for example, later on, then you have to take care of these things. But in early embryo, putting any cell anywhere, because there is no immune system developed that time. So it's not a problem. Yeah, go ahead. Anybody?
Hello sir, my name is Surendra Ghaskadbi. I am a student. Actually, I have three things to ask, but question is the last thing. So,
Whatever, you start with number one.
first is, actually yesterday night I was watching one of your YouTube videos, and in that the question that you over a decade ago, you gave your one kidney to Sarojma. Okay. And and you said that कि मैंने तो एक ही दे दिया but इसके बदले में मैं लाइफ टाइम के जितने भी गिफ्ट्स है वो सारे एक साथ दे दूँगा. So sir, I want to say that at that moment you just take my hat for yourself.
My wife always complains that I don't think of gifts. So jokingly in that podcast that you may have seen or some news item, I said एक बार ही दे दिया फिर अभी the slate is clean. Kidney दिया और क्या देने का? But that's true. Now we finished 12 years and both of us are happy and healthy. And our blood groups were different. Still that can be done.
Second thing is about your hobbies, sir. Actually, I find it fascinating your listening ability.
Oh my God.
You said that
That I learned in this hall only.
it is the hobby which you are going to pursue seriously in future like that. But sir, it's my request, if you took it seriously, my future may not sit here today over here. So, may I sir दिल से request है कि आप sir.
So quickly I'll answer your question. I used to whistle as a hobby. Then during COVID time, some students and friends said that why don't you whistle? So it was very quiet. Now there are so many dogs barking. There is construction going on. So I don't have a soundproof room. So I stopped. But that time they said very nice, sir. So all students are usually very sympathetic towards their teachers. So out of sympathy they said very nice. But it's okay. But I like whistling. During COVID times, I whistled a lot, and that's why I never had to use the oximeter because I could hold my breath.
Song itself करके आप, if it possible.
क्वेश्चन खत्म हो गया क्या? पहला वो पूछ.
Sir, I want to know that at this phase of life, what fascinates you? What are the questions on which you are stable right now or still okay?
So I don't have an active lab anymore because I don't have students. All the students have got their PhD now. So now my main what I enjoy is teaching developmental biology and science outreach. So I try to go to different schools. Like we went to a tribal school near Bhima Shankar a couple of months back, where I carry hydra. I cut them, show them reservation. Science outreach, there is so much ignorance. But one has to be very careful. Like the question was asked about religion. See, why do you want to promote science? You don't want to hurt anybody's religious sentiments because they are genuine. My wife is a believer, so everything goes on in our house. That doesn't mean that I should stop her, isn't it? But while communicating science, it doesn't help if you say that what you are saying is wrong. So, my current problem, actually, I am trying to overcome it, is to convey scientific facts and to try to, convince people about scientific facts without hurting their religious beliefs. Now, in case of evolution, it's very difficult. But somehow, you could.
Thanks.
Hello, sir.
Yes.
Sir, while in developing embryo, there is a mRNA gradient. In the developing embryo, in the presence of cytosolic streaming, why does not that mRNA gradient fades away and shifts?
So these mRNA molecules that I showed you, we didn't go into complex structures of that. They are actually tethered by some other molecules. They are both held there. So they are not free to move freely. Okay, so they are held with microtubules and microfilaments, and they are held at one place. It was not possible to show all this, but whenever they need to be released, the cytoskeletal elements will break off and the things will be released. So mRNA is restricted to different parts of the cell with the help of other molecules. So it's actually held together. Like our cell surface molecules are held by cytoskeleton. Likewise, that mRNA is also held. But once it is translated, then the protein can flow easily. That's how they form the gradient. But mRNA doesn't form the gradient. mRNA is at one place. Okay. Does that answer your question?
Okay, sir. Thank you, sir.
Okay.
Is there any question? Okay.
Yes.
Yes.
Sir, I have a question that with the rapid advancement in computational biology, how efficiently can bioinformatics approach and molecular docking be utilized to predict the map, how to map the complex gene regulatory network govern pattern information formation in early animal models?
So most of the work that I described so far was heavily based on bioinformatics because we compare nucleotide and amino acid sequences of different organisms, which is totally bioinformatics. We also predict three-dimensional structure of proteins, drug-protein interaction, and so on. But we have not come to a stage wherein we can completely predict the full map. That is because, see, bioinformatics and biology, experimental biology, will go hand in hand. Once you have bioinformatics tools, you do an experiment and find out whether it is the fact. Then feed that data back into computers. Computers will come out with a new model, which needs to be experimentally validated. So only bioinformatics without wet lab work is not going to help. Likewise, only AI will not tell you how we function, although that's the general impression. So ultimately, AI takes up information from us only. So that information we have to gather first.
Any questions? Okay, I have one question.
Yes.
I have read that the liver cells, hepatic cells, basically they do follow transdifferentiation. And you said that it is still not clear, very much clear about the transdifferentiation. Even though transdifferentiation process ends up with proper attainment of
Function.
organ, even shape also. And we are continuously failing to attain that shape while doing organ culture. So
If you understand how transdifferentiation takes place, then we'll have better tools to make organs in vivo. So transdifferentiation we have known at least for 60-70 years, which was shown initially in the eye, in the retina. Pigmented retina and neural retina were shown to transdifferentiate by some Japanese scientists. But exactly how suddenly one set of genes is switched off and the other set of genes is switched on, I don't think we have understood that. That's the problem. If that is understood, it will add to our knowledge of making organs in vivo.
thank you very much.